There are over 200 risk genes that are strongly associated with autism spectrum disorder or ASD. We know that these genes are involved in fundamental biological processes in the developing brain. A risk gene is a gene that is strongly associated with the likelihood of developing a particular disorder. We know that many of the autism risk genes play important roles in the development of excitatory and inhibitory cells as the brain develops. Therefore, if there's a mutation in an autism risk gene, this can lead to an imbalance in excitatory and inhibitory circuits or signaling. When we have this imbalance of excitatory and inhibitory circuits, this can lead to behavioral differences that we see in autism. My lab studies how risk genes that are associated with neurodevelopmental disorders, including autism, affect basic biological functions in the developing brain. Our goal is to identify basic molecular processes involving these genes at multiple levels, molecular, cellular, and circuit levels, and to use this knowledge to develop potential new pharmacological treatments. So in my lab, we use zebrafish as a system to study the function of risk genes in neurodevelopmental disorders and understand more about what these genes do in the developing brain. Zebrafish are a unique system because they allow us to see through fully transparent embryos and larvae, and this means that we can visualize basic neurodevelopmental functions in real time in a relatively simple, developing vertebrate brain. We can watch how cells in the brain divide, move, or fire, and we can study how these processes are impacted when risk genes are not functioning properly. In addition, we can use tools like molecular scissors like CRISPR to disrupt many different risk genes in parallel in zebrafish and ask how this impacts brain development at multiple levels: molecular, cellular, circuit, and behavioral. Zebrafish are an optimal system for studying how mutations in risk genes affect basic behaviors such as sleep or changes in responses to sensory stimuli, which are often affected in individuals with neurodevelopmental disorders. Because zebrafish have large progenies, it's possible to conduct high-throughput, small molecule screens in zebrafish larvae. And this allows us to identify new compounds that can potentially suppress or influence these behaviors. Our goal is for our research to lead to the development of potential new pharmacological treatments for neurodevelopmental disorders. We think that our approach really aligns with a precision medicine approach to neurodevelopmental disorders, where we can identify genes that are associated with risk for the disorder and then identify potential treatments that are relevant to mutations in those genes. As a practicing child psychiatrist, I realized there is a need for improved treatments for neurodevelopmental disorders, as well as a need to gain a greater understanding of these basic genetic and molecular mechanisms involved in these disorders.